Again, welcome to my channel. Today we are going to discuss about the hexose monophosphate pathway, that is the HMP pathway. Now, this HMP pathway, it is also known as the pentose phosphate pathway, which is a metabolic pathway parallel to the glycolysis, and the main role of this pentose phosphate pathway, it is to generate the NADPH and the pentose sugar, that is the ribose 5 phosphate. This pentose phosphate pathway, it is also known as the phosphogluconate pathway or the hexose monophosphate shunt. This pathway, no doubt, like that of the EMP pathway, this pathway it also involves the oxidation of glucose, but its primary role within the metabolism, it is anabolic rather than the catabolic. We know that the role of the EMP pathway, it is the catabolic to generate the energy. There is the glucose oxidation, and there is the generation of pyruvic acid. ATP generation, it is also carried out. But here within the HMP pathway, no doubt there is the oxidation of glucose, but within this, the different components which are synthesized within the HMP pathway, these are acts as an precursors for the different biosynthetic pathways, that is for the synthesis of the nucleic acids, for the synthesis of amino acids. So due to that, its role, it is anabolic rather than the catabolic. As it is known as the shunt of glycolysis, because majorly the main enzyme involved, that is the regulatory enzyme involved within the glycolysis or the EMP pathway, that is the phosphofructokinase enzyme, whenever it is inactive, then the glucose 6 phosphate, which is produced within the first step of the EMP pathway, from the phosphorylation of the glucose molecule, it is diverted or shunted into the HMP pathway, that is it is get introduced within the HMP pathway. Due to that, it is called as the shunt of glycolysis. This pathway, it is occurs within the cytoplasm of both prokaryotes and the eukaryotes. Majorly all of the reactions which are carried out within this pathway, these are divided into the two main steps or the two main types. The first four reaction within this HMP pathway, these are irreversible and oxidative, in which the glucose molecule, it is oxidized twice for the generation of two molecules of the NADPH, and then there is the conversion of the glucose into the ribose 5 phosphate, it is the 5 carbon sugar. It is an oxidative phase, and the next reaction involved within this pathway, these are non-oxidative. These oxidative reactions, these are followed by the series of reversible reactions, within which there is the sugar phosphate interconversion reactions, these are involved. Now here it is the reactions, these are given. Firstly, the glucose 6 phosphate, which is formed within the EMP pathway, whenever it is get introduced within the HMP pathway or shunted within the HMP pathway, then there is the activity of the enzyme glucose 6 phosphate dehydrogenase. That is the first oxidative reaction, where there is the oxidation of glucose, and there is the generation of first molecule of NADPH, and there is the formation of 6 phosphogluconate delta lactone. It is the first oxidative phase within the HMP pathway. After the formation of 6-phosphogluconate delta lactone, there is the conversion of this into the 6-phosphogluconate by the activity of lactonase enzyme. And then again there is the dehydrogenation reaction, it is carried out over the 6-phosphogluconate by the activity of 6-phosphogluconate dehydrogenase enzyme, and again there is the generation of the NADPH, means it is the second oxidative phase or the second oxidation reaction, within which there is the generation of NADPH2, and there is the formation of pentose sugar from the 6 carbon sugar, that is from glucose 6 phosphate, there is the formation of ribulose 5 phosphate. Now, majorly this ribulose 5-phosphate, it may be get epimerized to form the xylulose 5-phosphate, or there may be the isomerization of these ribulose 5-phosphate into the ribose 5-phosphate. Means there may be the activity of isomerase enzyme or the epimerase enzyme. Through the epimerization, there is the formation of xylulose 5-phosphate. Through the isomerization, there is the formation of ribose 5-phosphate. Now, these two molecules, that is the xylulose 5-phosphate and ribose 5-phosphate, these are combined with each other by the activity of transketolase enzyme, and there is the generation of a 7 carbon sugar, that is the pseudoheptulose 7-phosphate, and the glyceraldehyde 3-phosphate. Then from this glyceraldehyde 3 phosphate and the pseudoheptulose 7 phosphate, again there is the activity of trans-aldolase enzyme, and from that majorly there is the formation of fructose 6 phosphate and the erythrose 4 phosphate. That is, the from the pseudoheptulose, the one carbon it is transported to the glyceraldehyde 3 phosphate, and there is the formation of erythrose 4 phosphate, and from this 6 carbon again there is the generation of fructose 6 phosphate. Then from this erythrose 4 phosphate and the xylulose 5 phosphate, here it is the pathway where we can observe how there is a shunt it is formed in between the glycolytic pathway and the pentose phosphate pathway. Now we know that within the glycolytic pathway, the first step it is the phosphorylation of the glucose into the glucose 6 phosphate. Whenever the next enzyme involved within the third step of the EMP pathway, that is the phosphofructokinase enzyme, it is inactive, then this glucose 6 phosphate it is introduced within the HMP pathway, and there is the activity of the dehydrogenase enzyme. Means instead of formation of fructose 6 phosphate, there is the oxidation of glucose 6 phosphate into the 6 phosphogluconolactone. Again, within this, the ribulose 5 phosphate, these are get formed from the ribulose 5 phosphate, the ribose 5 phosphate, xylulose 5 phosphate, these are get formed. Within that, again, there is the formation of glyceraldehyde 3 phosphate. This glyceraldehyde 3 phosphate, which are formed within the HMP pathway, they may be reintroduced within the EMP pathway. There may be the reintroduction of these into the EMP pathway. Again, the fructose 6 phosphate, it is also get generated within the HMP pathway. It may be introduced within the EMP pathway, or it continues the EMP pathway. Means whenever there is the availability of this 6 phosphofructokinase enzyme, it may be fructose 6 phosphate, they may be again get entered within the EMP pathway from the HMP pathway. So due to that, it is also known as the shunt pathway. The glucose 6 phosphate dehydrogenase, which is the first enzyme within this hexose monophosphate shunt, is an important enzyme for the healthy cells, specifically within the circulatory system. The defects or the deficiency of this enzyme, it is leads to the reduced levels of NADPH, because majorly it is the major pathway for the generation of reducing power. An important reductive role of the NADPH, it is to maintain the glutathione in its reduced state. The reduced glutathione is a tripeptide which contains the cysteine. The reduced free sulfhydryl group, it is necessary to help the combat the reactive oxygen species often present within the cell. Now the one example of this, it is the erythrocyte. We know that the erythrocytes, these are lacking the mitochondria. So the major pathway through which there is the generation of the reducing power, it is the HMP pathway within this erythrocyte. Otherwise, within other cells which contains the mitochondria, within the mitochondria there is the generation of this reducing power. So these are depending totally upon the shunt for their supply of reducing power, that is on the HMP pathway. The low supplies of NADPH responsible to weaken the erythrocyte and making them more susceptible to the damage from the reactive oxygen species or the general oxidation. Now the significance of pentose phosphate pathway or the phosphogluconate pathway or the hexose monophosphate pathway. This HMP, it is the only cytoplasmic pathway which is responsible to generate the reducing power. This NADPH, it is produced in this pathway. It acts as a reducing agent during the biosynthesis of various molecules like that of the fatty acids. This pathway, it generates the 3, 4, 5, 6 and 7 carbon compounds, that is the glyceraldehyde 3 phosphate, erythrose 4 phosphate, ribose and the xylulose 5 phosphate, then the fructose 6 phosphate and the pseudoheptulose 7 phosphate. These are the different carbon compounds which are generated within the HMP pathway. All of these are acts as an precursors for the biosynthesis of the different molecules like that of the nucleotides which are synthesized from the ribose 5 phosphate. The pentose 5 phosphate pathway, it is very essential for the cells which are lacking the mitochondria, that is the RBCs, for generation of the reducing power. The triose, tetroids, pentose, hexose and heptose sugar are generated as the intermediate products of this pathway. This NADPH, that is the reducing power, no doubt it is utilized for the biosynthesis of various biomolecules Along with that, these are also used to reduce or detoxify the hydrogen peroxide within the cell. The resistance to the malaria in some Africans, these are associated with the deficiency of glucose 6-phosphate dehydrogenase enzyme because the malarial parasites depends upon the HMP shunt to reduce the glutathione in the RBCs. So here it is then significance of the pentose phosphate pathway. If you like this video, please subscribe my channel and also share this video with your friends. Thank you.